So Mark Daly is a really internationally known geneticist. He is an associate professor at Harvard Medical School, based at Massachusetts General Hospital. He's also the co-director of the Medical and Population Genetics Program at the Broad Institute. And at Mass General, he heads the Advanced Translational Genetics Unit. And what's really amazing about Mark is that he thinks not just about the genetics of autism, but the genetics of many different diseases, which allows him to compare insights that you can gain both from closely related disease, other neuropsychiatric diseases like bipolar, but also some from different sorts of diseases. And he's really made a tremendous contribution to our understanding of autism genetics. And so we're very lucky to have him speak today. Mark Daly.
Thanks, Chris, for the very kind and generous introduction, and thanks for the invitation to the Consortium retreat organizers to come and speak about where we've begun to get to in our understanding of autism genetics, particularly in light of the great technological advances in our ability to study the human genome that Chris alluded to that have been really emerging over the last few years. So, do I hit a switch or something?
Yeah, you just have to put a marker on presentation.
Oh, okay, great. Oh, we're getting there. Great. All right. Thanks very much. So I'm going to start back a little bit, and do I aim this at any place in particular? Or? All right, well, we'll just do this. That's fine. Anyway, the ultimate goal of the types of human genetics investigations that Chris and I and many of our colleagues in this community and internationally take on is really to give us some fundamental biological insight into autism or any of the disorders that we study in order eventually to learn about the diseases and the disorders so that we can study them better, but eventually so that we can develop much more intelligently therapeutics that are truly effective and truly addressing the root causes of disease. And as you can see, particularly from the lower panel, with respect to neuropsychiatric diseases in general and really almost anything to do with the brain, we have next to no novel therapies that have been developed and successfully implemented in clinic for nearly half a century. This is obviously, as was stated this morning, an unacceptable state of affairs. And primarily, if you talk to people at the pharmaceutical industry, arises from our fundamental lack of knowledge of where to attack. And that is what we hope genetics can begin to give us some insights into. Now, of course, we came through a tremendously exciting decade of research in the 1990s where, headed by Jim's work in Huntington's disease and other seminal efforts, Lou Kunkel's work in muscular dystrophy, the discovery of the cystic fibrosis gene in 1989, a tremendous ability to discover the underlying causal factors in what we call Mendelian diseases, diseases that are caused by mutations in a single gene that are severe and deleterious and that have really a deterministic relationship. If you get a mutation in this gene, you get this disease. Now, at the same time, down at the bottom of this slide here, there's this sad little blue triangles, and that tells us that over this period of time, we made next to no progress in any common or complex human disease in terms of our understanding. And why this was, of course, we didn't really have the tools in hand. And it's only been really now 10 years that not only the technologies have begun to become available for studying the genome, but we didn't even have the genome sequence except, about 10 years ago, let alone an understanding of how DNA variants, assort themselves throughout the genome. And then, obviously, ability to study the genome in the laboratory has come along slowly since that point in time. Progress has become much more swift in the last few years. First in the era 2005 to 2010, and these, if you've been for previous retreats over the last number of years, you've heard of a lot of different types of genetic studies that have provided us some fundamental insight into autism and many other neuropsychiatric diseases. through both an understanding of copy number variation, so places where there are small duplications or deletions of genes in the genome, as well as through common DNA variants, single nucleotide changes, some of which are in genes, some of which aren't, which are associated to disease risk. So copy number variation has been the focus, initially because we've been able to recognize it easily from early generation genome technologies. And starting with some seminal papers from Jonathan Sabat and Mike Wigler and Charles Lee's lab, we've begun to appreciate not only that large deletions and duplications exist in the genome, but that they actually are found much more often in cases of autism than in controls from the background population. In 2008, the key finding from the Autism Consortium was one of the first published where a specific defining risk factor was identified in a small number of cases, that being the 16p11.2 deletions and duplications. And there have been, since this time, a number of different studies that have emerged identifying a number of different regions in the genome. But generally, here are a listing of some of them, but generally these studies have concluded that most of the individual variants that are found are unique to individuals and families and not seen recurrently very often in the population sample. And so while there's certainly an elevated risk to carrying deletions and duplications, they don't easily necessarily point us to a small number of regions in the genome. And inescapably, this tells us that there are hundreds of genes, hundreds of regions of the human genome that have critical genes involved in brain development or other functions that are relevant to autism risk, and that when you delete them or duplicate them, you increase the risk of autism. So we know early on from these studies not too much about specifics of the biology, but we really begin to see that there's going to be a large number of genetic contributors. And the other things that were fascinating and emerging from the research at this point in time is that these copy number variants, while individually rare, they begin to be seen across a number of different disorders. And so, remarkably, nearly every copy number aberration, a duplication or deletion, that's been identified as associated to schizophrenia risk is also associated to autism risk. And this presents now genetic and biological links between disorders that we may not have necessarily appreciated, or some of us may have appreciated and others may have debated for a long time, between disorders that we've considered clinically distinct. And this is, of course, telling us that the biology of these disorders, which may happen at a very fundamental point in time in brain development, is not respecting our traditional clinical boundaries, and in fact only has a looser rather than a deterministic connection, as we saw in those Mendelian diseases. So here are some of the lessons that we took away, and these are important lessons both for their individual insight into autism genetics, but also in terms of calibrating our thinking for what we're going to learn when we start sequencing entire genomes or exomes, as Chris was describing. So we moved on from these early studies of copy number variant segments of the genome to look more generally at common DNA variation. And of course, the consortium was very much out front in supporting an early genome-wide association study that was published in 2009, but more importantly, made the raw data from the research study available a year before the research itself was even published. And so researchers throughout the community began to benefit from this type of research. And this set a model that the field has followed over the last few years. And now, rather than having separate silos of research involving families collected by the Simons Foundation, families collected by AGREE, families collected by the Autism Consortium, all of the geneticists are collaborating together to pool results. And this is from a publication of one of those pooled studies done in schizophrenia. The case and control samples are much, much larger, which is one thing we may have to address in autism, but it's also been published, so it's easy to point to. And one of the key reasons I point to this is because some of the things that we're learning from the genetic studies of schizophrenia are turning out also to be quite relevant to autism risk as well. And so these types of studies are finally, for the first time this year, because of the collaboration of researchers throughout the world, providing some direct and key biological insights for the first time into the pathogenesis of autism and schizophrenia. And there's a particularly exciting pathway that's been emerging in the schizophrenia study that's shared with autism study from the unpublished data that I can describe to you, in which a microRNA and a key gene that's regulated by that microRNA in a different part of the genome are both strongly associated to schizophrenia and autism risk. And so like the CNVs, these types of common DNA variants, while they don't cause disease in a Mendelian fashion or in a deterministic fashion, tell us about what the biological risk factors are and give us some pointers into specific processes that underlie disease. So we've been seeing now in the last year that these types of studies of common DNA variation, while they generally are of very modest impact on the individual and even very modest impact on the gene in which they occur, are beginning to make some discoveries in schizophrenia and even now in autism. But unlike many of the other disorders that are studied by geneticists, whether it's inflammatory conditions such as Crohn's disease and colitis, type 1 or type 2 diabetes, or even the genetic studies of height and body mass index, the numbers of samples that have been aggregated for investigation in neuropsychiatric diseases, schizophrenia, autism, and related disorders, are much, much smaller. And we as a research community need to work hard to make sure that these diseases, which are so important to us, do not fall by the wayside in medical research as they may have in previous years, simply because the investment's not made in doing the studies. And that's where I think the critical contribution of the Autism Consortium in really investing in working with families, working with clinicians to empower them and to really generate new research programs coming out of that work is critical. So we learn many of the same things and begin to get some specific biological clues from these types of genome-wide association studies, as I've described and many others have described in the past. But really, we've only scratched the surface of the beginnings of our genetic understanding of autism and many disorders from the technologies of 2005 to 2010. And so what everyone has been so eagerly awaiting has really come to pass in the last 1 to 2 years, and that's the advent of the ability to sequence entire genomes and really collect the full genetic information, where then we become no longer able to complain that, well, it's just in the part of the genome or that we haven't looked at yet or we can't sequence the whole genome. Well, now we can. So if we can't start making progress in the genetics of our diseases, it's on us, or at least it's on our ability to creatively think about how to analyze the full genome sequence. So if we survey what's the type of research that's been published from whole genome or whole exome sequencing studies in the last year to year and a half, it's really clustered in a couple of critical areas. One is, as with those old studies in the 1990s, the genome sequencing has allowed us to make really rapid progress in a number of extremely rare disorders, where by sequencing the genome, we can pinpoint the one gene that is disabled in the two families that share this extraordinarily severe and rare genetic condition. And we haven't really had the technology to do that before, and it's of critical importance. It may have limited relevance beyond extraordinarily rare conditions. There have been a large number of review articles that don't have any new data content but tell you how important genome sequencing is, and actually we have almost too many of those, if you ask me. There's been an impact of next-generation sequencing in cancer studies and obviously a whole host of new analytic methods. But what there's been very little of in terms of discovery thus far is in novel findings in more complex and polygenic diseases. And this is really, telling us that there's a fundamental challenge not in collecting the data, but in really figuring out how to learn from it, interpreting the genome sequence data. And that's because some things, when you sequence the entire genome, are very easy to ask. Like if we can ask, how many new mutations do you have in a protein coding gene? The answer is, every one of us carries about one. Not so complicated. Can we ask if there are 2 disabling mutations in the same gene? This is a very, very rare occurrence anywhere in your genome. So these are analyses which can be very informative because you're not clouded by a large number of background DNA variants arising. But most of the complex diseases, as I've said, we study, involve hundreds of contributing genes. And if you actually sequence the entire genome, even just looking at the protein coding segment of it, there are 20,000 DNA variants that each of us is carrying around, including 100 or hundreds that are extremely rare in the population. And so you might look at them and say, well, that could be exciting, but then there's one a few genes down that also could be exciting, and then so on and so forth. So we really need to focus on collecting large numbers of patients so that we can begin to understand and interpret the DNA sequence as we find it. And so we've been working for the past 2 years with an NIH-funded collaboration that's supported by both the Mental Health Institute and the Human Genome Institute. And you see a number of the investigators here. And the two types of studies we've done so far are sequencing the exomes, which, as Chris said, are the 30 million base pairs of DNA, so still quite profoundly large, that encode for proteins in almost 1,000 cases and 1,000 controls, as well as sequencing the exomes of 174 trios, which is just our genetics vernacular for 2 parents and an affected child. So this will be the study that I'll briefly describe for you. It's still obviously a work in progress, but in particular, we focused locally on the sequencing of trios, 2 parents and an affected child, from 104 of the participating Autism Consortium families. So the fundamental challenges, as I've outlined them for you, are, of course, that there's many genes involved, and so we don't really expect very many patients to carry mutations in any one particular gene, which, of course, then makes it very, very hard to find them in the first place. And the reason it's hard to find them is because when we do this type of sequencing study, we find hundreds of thousands of DNA variants scattered throughout the 20,000 protein coding genes in your genome. So what do we begin to find with the most detailed genomic look at patients and families that has been performed to date? The existing screening data that's been collected by the consortium and nicely arranged in informatics databases supported by the consortium that all of our research community can utilize points to the fact that 8 of the patients that we're sequencing carry a known or interpretable karyotypic abnormality or a large deletion or duplication that we consider relevant to autism risk. And 2 of those are 16p11 cases. The other ones are all single cases of different regional abnormalities in the genome. So remember, this is out of 104 patients. So clearly, there's a contribution from these larger genomic aberrations, but it's only in a small number of cases. And this will be sort of a recurrent theme. So another area that we've begun to focus on, and Chris will, I expect, pick up this theme because he's really spearheaded a lot of the research into trying to find recessive subtypes of autism. And recessive subtypes simply refers to things like cystic fibrosis, where if you have 2 copies of a gene disabled, each the maternal and paternal copy of a gene, then essentially you can't make that protein. And this, if it's an important gene, is clearly a high-risk scenario for whatever outcomes that gene is relevant to. And it turns out that if there were recessive acting subtypes of autism, we would not have seen them in any of the previous types of genetic research. They'd be sort of hiding in and amongst many other causes. And so we begin to look for these in the data, and we actually do find some evidence for these, like the copy number aberrations. It's a small contribution, maybe 5% of the cases that we're seeing, but clearly this is beginning to point us to some specific genes that may be involved in autism risk. And sometimes we come across instances where some of these might fall into the category of some of the things that Pearl was describing, where if we look through the literature, we find that, gee, other people have reported loss of function mutations in this gene as having specific phenotypic outcomes, maybe that are related to autism or the diagnostics of autism. And this may be instances where we need to start going back to IRBs and considering what we might informing families. The other area in which the sequencing of 2 parents and a child is informative is that we can find, by comparing what is present in the DNA of the child that's not present in either parent, the identification of DNA spontaneously arising DNA mutations. These may have a high impact, and because they're not transmitted from generation to generation, they may have a severe phenotypic impact, but not be required to be passed on throughout human history, as most of the common DNA variants are, and so could have a bigger individual impact on a gene function. But of course, the challenge is that these actually occur in everybody. Nearly all of us walk around with about one spontaneous mutation hitting a protein coding gene somewhere in our genome. And so again, we have this difficulty in sorting out what's relevant to an individual case and what may be simply the background rate of mutation in the genome. So when we start to look at this from a more statistical standpoint, we see that maybe there's a few more of these types of mutations in our cases than would be expected from the mutational process at random. It's not a terrifically convincing excess, again, maybe landing somewhere around 5 to 10% more of these types of mutations in cases than you would expect by chance. And there is an excess that's modestly statistically significant of those mutations that are particularly severe, these ones that knock out a copy of a gene. And so certainly we begin to see, as with the CNVs and as with the recessive cases, an impact of spontaneous DNA variation as well. And again, as with some of the previous types of studies, we can partner with other researchers, and we are doing that again in a very aggressive and time-sensitive fashion to see what insights can come out more quickly. And by combining the results of what was found from this study with several other researchers who are performing similar studies, we can get to a much larger number of trios sequenced and find that, well, we now see some genes that are being hit twice, one in our study, one in another study, or what have you. And these, we're seeing more of these than we'd expect by chance, and these begin to really point us to some specific subset of the genes that are relevant to autism. But as again, you can see, it's a small number of cases overall. We can also begin to take the genes that are discovered from this as having spontaneous mutations or having potentially recessive acting mutations and ask whether those genes point in a harmonious fashion to specific pathways and processes. And so this is where we try to draw in more sophisticated information from other areas of biology, such as detailed protein-protein interaction networks and so forth. And we can find that there is some statistical evidence for certain pathways and processes being clearly enriched in the genes that are being hit. And this begins to get us a little bit closer to our ultimate goal of informing on the underlying biology of autism. And so what we've learned as a whole from autism sequencing to date from this last part, we clearly see that there are an excess of these type of de novo mutations. Those are the ones that were spontaneously arising. These don't appear to cause disease, as in, for example, cystic fibrosis or Huntington's disease, as we're familiar about thinking about Mendelian disease, because we do find these types of mutations also in control populations. And now, across many NIH-funded studies, more than 12,000 of these exomes have been sequenced across a number of different clinical areas. But the other thing we know is that the majority of the cases don't have a simple genetic risk factor underlying the diagnosis of autism. And so we put this in a slightly more graphical form. The numbers may move around a little bit as we sequence more and more families over the next few years, and we begin to get a better understanding of how to interpret the data from a whole genome sequencing experiment. But it's clear to us that while there are important contributions from chromosomal abnormalities, copy number variants, from recessive models, from spontaneous point mutations, it's clear that the majority of cases don't have these types of simple genetic underpinnings, as well as the fact that whether you have one of those types of genetic events or not, nearly all of the cases of autism have a high-risk inherited genetic background that we've only now just begun to learn how to study and to draw biological clues from. And so we are certainly encouraged by the fact that all of the avenues to investigating genome sequence are beginning to provide some fundamental insights into the biology of autism. But for all of these different categories or bins of genetic variation types, we've only just begun to identify a few of what's a much larger pool of genetic risk factors underlying autism. And if we can't drive these studies to their natural completion, we're not going to have the complete picture. And so I hope everyone is both encouraged, but encouraged to press on further with our genetic understanding, and accepting that this is only a down payment of sorts on what we can ultimately achieve with genetics. I look forward to coming back again sometime over the next few years and describing how this has progressed further. So with that, I thank obviously many people, but most of all, this wouldn't happen without the dedicated efforts of both the families and the clinicians who make this research possible with the support of the Autism Consortium. And so I thank them most of all. And I'd be glad to take a few questions, I guess. Sure. Yes. I'm sorry, I can't hear you. Can someone turn that